Hi everybody, I'm doing this lesson as a flipped classroom. My goal is to give you the notes while you're home so that you can be writing this in your notebook. Then when we're in class, you'll have more time to spend on problems and I'll be right here to help you through it. So the first unit that we're doing this for is on cells. To begin, we want to talk about the organelles that are most commonly found in cells. We'll start with the membrane. The membrane is the exterior portion of a cell that allows certain molecules to travel in and out of the cell. It is made up of a phospholipid bilayer, so two layers. When we did chemistry, we talked about how the polar heads would face out and the tails would face in. This is our phospholipid bilayer. Proteins can also be embedded in that bilayer. So we're going to draw a picture together, and here are those polar heads. Here are the fatty acid tails, and here's the second layer. We can label this as the outside of the cell, and this is the interior of the membrane. There's a couple other molecules that we should also show in our membrane. First of all, we want to show some cholesterol. Remember, cholesterol had the 4 carbon rings? That helps spread out the tails a little further and give the membrane a little additional flexibility. There are also membrane proteins. So I'm going to draw a large protein in this space. We're going to go through a lot of information about the membrane as we get further into this unit. So for today, I just want you to realize there are proteins embedded. I'm sure you probably remember from Regents Bio, some of these will make great channels for certain molecules to go through. And we'll just add a couple more. We'll add a couple more phospholipids here. You'll also remember that word amphipathic. These phospholipids are amphipathic, with the heads being polar and facing the water, very hydrophilic, and the tails facing inside because they are nonpolar and very hydrophobic. This structure allows a membrane to be selectively permeable, which means that only certain molecules will be allowed to travel in or out of the cell. Things that travel very well are small, uncharged molecules. And the reason why uncharged works well is because this is very nonpolar in the center. So for something to actually slip right through the membrane, something like oxygen that is small and nonpolar can go through very easily through a lipid bilayer. Water can go through a lipid bilayer, but not in great amounts. In areas of your body where you need a lot of water moving, like in the kidneys that are filtering water, there are special proteins called aquaporins that will allow water to transport easily through. If things are larger, like proteins and sugars and nucleic acids, or if they have a charge, like salt, they may be able to enter by another means, which means they might go through a membrane protein or by endocytosis or exocytosis. You might remember diagrams like this, where an amoeba would surround and engulf a molecule. This would be my amoeba, and this would be the food, and that would be an example of endocytosis. So that's how larger molecules may enter or exit. The next organelle that we want to focus on is the nucleus. So the nucleus, you usually hear about it as the control center of a cell. And the reason why it's considered a control is because the information to do all cellular activities are enclosed within our DNA. DNA is stored and replicated in the nucleus. DNA is the directions for building a protein. So all the directions for building any kind of protein, any kind of enzyme, any kind of structure that that cell or the organism needs is contained in that DNA, and that is within the nucleus. The nucleus is the site of transcription. Transcription means copying the DNA into RNA. If you remember from biochemistry, RNA is only single-stranded. It has a much easier time leaving the nucleus. DNA often stays right in the nucleus. So site of transcription, which is the production of RNA using DNA as a template. Another thing about the nucleus is that it has a double membrane. The double membrane is like a backup protection to keep the DNA in a safe, secure place. There are pores in the nuclear membrane, and the pores are small enough to keep the DNA in, but let RNA out. All right, before I do the next organelle, I also want us to start a diagram. So your notes are in one place. You might want to take a separate page of your notebook to draw a cell. So we will start out just drawing a pretty typical animal cell. I want to have a large membrane here, and I can label this my cell membrane. Then I want to take and do another circle. I'm going to double enclose it. This is my nucleus. You can go ahead and put some DNA inside of it to show that that's where the DNA is contained. And we can also label this the nuclear membrane. And that contains pores or little openings where RNA might be able to get out. Now if I go back to the notes, the next organelle, the third one of the day, is mitochondria. I'm sure you remember it being called the powerhouse of the cell. Powerhouse just means this is where the energy is made. Energy in the form of ATP. So how do we get energy? How do we get ATP? By breaking down sugars. Cellular respiration is the breakdown of sugars into carbon dioxide and water. And through that process, a lot of energy is released, a total of 36 ATP per glucose. So there's our formula for cellular respiration, glucose plus oxygen, why we constantly need that intake of oxygen, so that we can, I always give you that as an example, blowing out, you're breathing out carbon dioxide, and you can feel the water vapor that you're breathing out as well. And there's the energy production occurring. So this reaction happens in every cell of every living organism within the mitochondria. If you have a part of your body or a part of another organism's body that requires more energy, chances are it's going to have a higher content of mitochondria within that cell. So for example, muscle cells have way more mitochondria visible than a blood cell. Mitochondria are another organelle that have a double membrane. The outer mitochondrial membrane is the outside, and then the inner membrane is very highly folded. So when we draw it, I usually tell people think of it like a hot dog, and that squiggly line going down the middle is the mustard. That is the cristae. The cristae is the inner folded membrane, and that's where a really important part of the cellular respiration occurs. So we want to go back to our diagram, and we want to add some mitochondria. So long and thin like this, inner folded membrane. And you can draw a couple of them around and label it mitochondria. And you can label that inner membrane the cristae. On to our next organelle, ribosomes. Ribosomes are the smallest organelle within a cell, and they have no membrane of their own. They are simply a structure that is protein and ribosomal RNA. So they're a nucleic acid protein combination. This is where proteins are made. This is the site of protein production. We call that translation when we go into molecular genetics and we study DNA and how proteins are built. It's going to really be so dependent on ribosomes. The directions that originally came from the DNA get transcribed into RNA, and then the RNA leaves the nucleus and goes to the ribosome. And at the ribosome, amino acids get put together in a very specific order. If you do remember back in Regents Bio, there are codons, 3 letters in a row. So for example, AUG. AUG codes for methionine. Methionine is one of the 20 amino acids that your body has. So it will assemble the primary structure of a protein. You can find ribosomes free, which means they're just loose in the cytoplasm, or they could be attached to ER. ER stands for endoplasmic reticulum. Before we draw and add this in, we're going to go ahead and do the next organelle, which is our ER. ER stands for endoplasmic reticulum. ER is a transport system. It's literally a system of tubes that run through the cytoplasm. Those tubes could be smooth or could be rough. If they are rough, they actually have ribosomes right on them, which gives us a lot of information about what they do. If the ribosomes are on it, they are going to be responsible for moving the proteins that are built at the ribosome. They'll move it to the next station within a cell, which is called the Golgi bodies. We'll do that one next. If it is smooth ER, the endoplasmic reticulum has no bumps on it. It has no ribosomes, and they often have a different job. They can actually transform other molecules into lipids to build the lipids that cell or your body needs. They can also store ions. That'll be important in a muscle. They can store calcium ions. Detoxifying chemicals found in cells is another job that they believe ER of the smooth variety does. So let's go back to our diagram here. And what we want to do is just draw a nice system of tubes coming out from the nucleus here. Ribosomes, so this will be my ER. I'm going to put some ribosomes on a section of this ER. And I'm going to label this rough ER. And I'm going to label this a ribosome. I'll also draw some others and label those. So these are free ribosomes. These are attached ribosomes. You will find both in most cells. This section over here where there aren't any ribosomes, we'll label that smooth ER. On to our next organelle. A vesicle is simply a membrane that forms off of, oh, we need our Golgi bodies. 5. All right, 6. I'll do the vesicle after I do Golgi bodies. I just got to create a new screen here. So we'll say number 6, Golgi bodies. The job of a Golgi body is that it packages proteins Alright, there we go. Golgi bodies packages proteins from the ER, the endoplasmic reticulum, to be transported in the cell or to actually leave the cell. When the Golgi body packages up these proteins, it's literally just encasing them in a membrane so that they are protected and they're sort of marked. And by marked, what I mean is they can go to another piece of membrane, fuse with it, and dump their contents out of the cell. So we can go back here to our diagram, And I'll use another color. These often look like a little stack of pancakes with dripping blobs coming off of them. So I want to go like this and have a couple things coming off. These are my Golgi bodies. And what's coming off of them is a vesicle. The vesicle is that enclosed membrane formed by the Golgi bodies. And now that vesicle could fuse and this could leave the cell. So if we think about this for a minute, just put it together a little bit. In the nucleus we have our DNA. That's our directions for building a protein. The directions can't leave, but a section of gene could be made into a strip of RNA. That's called transcription. That RNA can then leave the nucleus through that nuclear pore, go to a ribosome. The ribosome uses the RNA to actually build the protein, put together the amino acids in the right order. So here is my protein that has been built by the ribosome on the rough ER. That protein will then travel through this system of tunnel to the Golgi body and eventually be packaged. So there's my protein. Protein that was built, maybe it's a hormone, maybe it's insulin. Maybe this is a cell in your pancreas that produces insulin to lower your blood sugar level. That protein can now, because it's in a vesicle, fuse with the membrane and be released out of this cell. All right, when we go back to this one here, I realize now this numbering will have to change. So our 7th organelle will be our vesicle. Really simple, that was just our membrane encasing our protein. So formed of membrane from the Golgi body or the cell membrane. Contents may be proteins or lipids if it's coming from the Golgi bodies, or dissolved food if it's coming into a cell. So a vesicle could also be formed the opposite direction. Let me go like this and just quickly show you what I mean. Let's say, oops, don't mean to move that. Let's say this one right here just came into the cell. If this is coming into a cell, we would also call it a vesicle because there are contents and it is enclosed in a membrane. So the term vesicle truly just means contents enclosed in a membrane. Vacuole, we'll make this number 8. Vacuoles are storage organelles. They could hold extra food, they could hold waste. In a plant cell, a vacuole is very large and it's filled with water, and the water exerts a pressure. I always go like this. That pressure is called turgor pressure, and it pushes the water against the walls of the cell membrane, which push against the cell wall. So that allows the shape of a plant to stay upright. There is no bone structure within plants. They have cellulose in those cell walls, but it's really the pressure of the water filling up the cell and pushing against the cellulose that gives it its shape, which is why if a plant dehydrates it, it wilts. And it wilts because it's losing its structure, it's losing its shape, it's losing the water that pushes against the walls. So what I want to do, I want to go over here, and we could just draw a vacuole. There's nothing really special to draw about it in an animal cell. Just label it a vacuole. But what I want to do is create a plant cell. I'll use green. I'll write plant cell here. You might just be able to do this in the Cornell section to the left of your notes there. Here's my very cellulose cell wall. A cell wall is not alive. It is simply fibers that surround each cell. The living portion is where the membrane is. There is usually a very large vacuole. And the vacuole exerts pressure. I'll put TP, turgor pressure. The turgor pressure that it exerts on the cell helps hold the shape of the plant cell. All right, we'll move on to our next one. This would be number 9. Lysosomes and peroxisomes. On a diagram, it is often difficult to tell if something is a vacuole or a lysosome or a peroxisome if it's in an animal cell, because they tend to be small, they tend to be circular, also any vesicle, and it's really difficult to tell what one is. So instead, we focus on what are the jobs that they do if I'm told this is what it is. A vacuole in a plant cell will be easy to notice. A lysosome and a peroxisome are all about digesting. They help break down substances. So they contain hydrolytic enzymes. Remember hydrolysis, adding water to lyse, to break something apart. That's going to be important for intracellular digestion. So let's say that amoeba that we drew a picture of before takes in some starch. Starch cannot be used. It has to be digested first. Cellular respiration uses glucose, and you can get glucose by breaking down starch. So when the starch comes in, it would form a vesicle, and there would be the starch inside. A lysosome, I'll do it in blue, would fuse with this, and their contents would be broken down until it was glucose. And then that glucose could be used by the mitochondria to build ATP. So, lysosome contains enzymes to digest larger molecules that are coming into the cell. Could break down food, could break down toxins. Peroxisomes break down H2O2, hydrogen peroxide, just like catalase, the enzyme that we used in our last experiment. When cell parts have gone bad and need to be recycled or regenerated, this is also the part of the cell that will break it down. Your red blood cells only live about 6 weeks, so your body is constantly reusing those cell parts, breaking them down using lysosomes within individual cells. Number 10 is what we call cytoskeleton. Cytoskeleton is the protein fibers that help give the cell shape and structure and support. They're usually connected to the cell membrane, so you will see them along the outside of the cell. They may extend to the exterior, meaning they might actually go all the way through the membrane and go out of the cell. When they go out of the cell, they're usually to help the cell move. 2 really easy ones to think of are cilia and flagella. Cilia you see on a paramecium. A paramecium is an organelle that looks a little bit like a slipper, and the tiny hair-like projections that are around it are cilia. A flagella is a big long tail. That tail-like projection is made up of a protein. It goes all the way from inside of the cell to extending outside of the cell. There are some organisms that are dinoflagellates, which means they have 2 flagella attached to them. There are some that just have 1. Centrioles are another example of cytoskeleton. Centrioles help chromosomes split. When a cell is going to replicate, go through mitosis or meiosis, all of the chromosomes double first, and they look like this. One chromosome doubled, looks like an X. We call them sister chromatids. Well, those sister chromatids are connected at a centromere, and they need to be separated to the opposite poles. So there is a region, it's called a kinetochore region on the centromere, where a centriole's spindle fibers would attach and pull the chromosomes apart. So centrioles found in cells help chromosomes divide and go to opposite poles during mitosis and meiosis. All right, so let's go back to our picture. And let's add some of these things. We could add a centriole here. Centrioles almost always appear on one side of a cell and there are two of them, and they're aligned perpendicular to each other, and I draw the lines to show the protein strands that are inside it. They often have a particular arrangement, like a 9x3 arrangement, which means 9 triplets, so 18 different protein fibers in that arrangement. So we'll label this a centriole. But then I also want to have just some others around the outside here. These are microfilaments, filaments of protein that help give this cell its shape. So we will call this the cytoskeleton. And those microfilaments are the protein strands that help give this cell its shape. We already drew cilia and flagella as an example, so I won't attach any to this cell. We have one organelle left. It is only found in plants. It is chloroplast. So this would be number 11, our final organelle of our notes for today. A chloroplast is only in a plant. It also has a double membrane. The outer membrane plus an inner stacked membrane that's called a thylakoid membrane. This is a synthesizing organelle, which means it builds things. It's going to build glucose, and it's going to use carbon dioxide and water. The thylakoid membranes contain chlorophyll. That's what gives it the green color. Chlorophyll is a protein. It is a pigment that's capable of absorbing photons of sun energy. When it absorbs that energy, it allows hydrolysis to occur. Hydrolysis is the splitting of water and the beginning of the steps of the light reactions of photosynthesis. Those reactions are eventually going to lead to the building of glucose. So right here in this diagram, or in this section of our notes, we'll draw a chloroplast, and then we'll add some chloroplast to our plant cell diagram. So a chloroplast, whoops. A chloroplast has an outer membrane, and then it has stacks of inner membranes. So make some stacks like this. The stacks are often connected. The stacks themselves are the inner membrane called the thylakoid membrane. And I could label some chlorophyll. There's also a fluid portion, a liquid portion. We call this the stroma. S-T-R-O-M-A. That is the liquid portion. And this is my outer chloroplast membrane. So this is one chloroplast, inner membrane, outer membrane, and then the liquid portion of it, the stroma. The chlorophyll is found within the membranes that make up these stacks, these thylakoid membranes. The thylakoid membranes, or the stacks completely, are called a grana. So we can take this whole section right here and label that grana. So there are the grana, and there's the liquid stroma, and each membrane of the grana is called a thylakoid membrane. It is a phospholipid bilayer, just like any membrane that we've done before. The only difference is it has that unique molecule chlorophyll found in it, which is why we only find it in plants. So if I go up here to my plant cell, and I still want to be inside the cell membrane, so this was my cell membrane. I want to have a couple chloroplasts within my cell membrane. Not drawing this very good, but here's my little stacks of membranes inside. I could also finish off this diagram and add a couple more just as a refresher of the organelles that we did. A mitochondria because plants do respiration as well. Some ribosomes, some ER, some Golgi bodies, a vesicle. We already have a vacuole. Lysosomes and peroxisomes are not as likely to be found in a plant cell because they don't eat. They make their own food. So our animal cell, we could label some lysosomes and peroxisomes, but it's not as likely that we would find those in a plant cell. You would still find some peroxisomes because hydrogen peroxide is a byproduct. If I go back to the animal cell diagram, I think I probably just left out those lysosomes or peroxisomes. All right, so that concludes the organelle portion of the notes. Please feel free to rewind, go back, stop it, look it over. I also on Moodle have several things for you to look at. There are websites that you can check out that have articles, diagrams, animations. What I do expect is for you to be able to link organelles to each other. How do they work together as a system? Thanks.